Okay, so thank you, Alain, for organizing this meeting and for putting together this very nice session. We're interested in the similar questions that Tommaso introduced already, and I will try to make it a little bit more broad. We're interested in how early experience shapes brain function. This is a common theme. If you go around the brain, you'll find that there are moments of time where genes and environment interact potently in so-called critical or sensitive periods. And our interest has been to understand what is the timing mechanism, because we think it's very important that these windows of plasticity come on, last, and then close with particular sequence. In fact, if you look at critical period timing throughout brain function, you'll see that there seems to be a hierarchy or a cascading nature of these, where primary sensory modalities feed into motor function and then higher cognitive functions. And so we suspect, and this will be a theme through this talk, that neurodevelopmental disorders, mental illness in particular, may reflect in part mistiming of these kinds of windows early in life. And so a theme that we've been pursuing over the years, and which will become the bulk of this talk is the notion that this is played out at a local circuit level as a balancing act between the excitatory and inhibitory circuits in the neocortex. That's where we focus. The excitatory cells, of course, are the overwhelming majority there, the spine-bearing neurons, as you just heard, and the inhibitory neurons quite heterogeneous in their morphology and connectivity. And in particular, we suspect there's one cell type, the parvalbumin-positive basket cells you've been hearing about, as being fundamental in orchestrating these windows. Usually when I give this talk, and many of you have heard me give this talk here, I start with the visual system, but thanks to Alain's brilliance, I don't have to do that today. Tommaso has done it very well. So I'll jump to the other extreme. To illustrate the importance of sequence and critical periods having cascading effects, let's take the very complicated example of autism. Autism is thought to be a disruption of a variety of brain functions. And it might start with primary sensory issues or hypersensitivity, for example, but there are many complex cognitive features like language and so on, emotional brain, social brain, and so forth, which need to be orchestrated in order for proper human behavior. And so I'm going to jump to a part of the brain that is very important in integrating a number of these kinds of features, and use that as a point of illustration. So the insular cortex is a higher order associational area in the brain, which is responsible for integrating sensory modalities as well as visceroceptive signals from inside the body, and has been implicated in a number of human imaging studies in these various features. Very complex functions such as language, pain perception, emotion regulation, and empathy, social interaction. And in fact, if you look at the brains of autistic subjects, this part of the cortex typically shows differences in social tasks versus non-social tasks, for example. And so we thought it might be interesting to look at the development of insular cortex in an animal model and see if we could pull out some interesting defects that we could pursue in mouse models of autism related to multisensory integration. Just to remind you, insular cortex is called insular cortex because it's tucked deep here in the human brain and is thankfully quite accessible in the mouse brain, right here on the surface, quite lateral. And we can apply intrinsic signal imaging techniques such as flavoprotein imaging of mitochondria and neurons to directly look at this insular cortex, which sits just anterior to the auditory fields in the mouse brain. So the mouse is tilted on its side a little bit, and we image the insular cortex here, which we can identify repeatedly thanks to the blood vessel landmarks. And because it's a multisensory brain region, we can apply different stimuli such as tactile stimulus, an air puff to the front paw, or a tone to the ear. If you look at the activation of this region, you'll see that touch is a potent activator. As I told you, it lies just in front of the auditory field, and so you see there's an auditory activation here anterior to the large auditory cortical area. And then you can, of course, combine the two stimuli and you get a multisensory response. And so you can plot that as the change in fluorescence due to touch, tactile, auditory, or audio-tactile. And what you'll see in C57 Black 6 mice as adults is there's a super additive effect if you stimulate both together. And if you just focus on the black traces, you see these across a number of sound frequencies. Mice, of course, have a broader hearing range than we do. If you look at a mouse model of autism, such as the inbred BTBR line, you see again that there is a nice tactile response, an enormous auditory response, and as a result, very little super additive effect of combining the two stimuli. And this result holds up across a number of sound frequencies. And so clearly there's an impairment here in these animals which have outwardly very odd behaviors, like repetitive grooming behaviors. The insular cortex has been implicated in the control of urges, for example. So what happens inside the insular cortex? Let's see. Why is it not going? There we go. So if we look at the inhibitory markers, for example, we find that inhibition is immature in the insular cortex of BTBR mice. GAD65, a presynaptic label for these GABAergic neurons, parvalbumin, as you've heard about, making these inhibitory basket-type synapses around target cells labeled with nu-n, and the perineuronal nets all show impoverishment in the BTBR mouse. This is quantified here and also functionally relevant. If you record miniature synaptic events, you find that the frequency of inhibitory events is severely reduced in the BTBR mouse. And so we could rescue this kind of weak inhibition using pharmacology. If you give benzodiazepine type drugs, you can prolong the duration significantly for these miniature inhibitory events and enhance the impact of this weak GABA input. If you enhance inhibition in the BTBR mouse, this lack of superadditive multisensory integration acutely recovers. So this is under anesthesia, improving inhibition with the drug right on the table. Instead, C57 Black 6 mice, which already have the superadditive effect, adding more inhibition actually impairs their multisensory integration, and this washes out as the benzodiazepines wash out. So given this pharmacological ability to restore EI balance, we tried to determine whether we could rescue this multisensory integration with a developmental perspective. And the reason for that is we noticed in young wild-type mice, C57 wild-type mice, that the auditory response actually is fairly pronounced at young ages, just after hearing onset. And that there's a developmental pruning process. This is a particularly severe example where the auditory response is reduced over time. In the BTBR mice, the auditory response fails to be pruned. It remains large. The insular auditory field remains large. And as a result, concurrent with this pruning of this auditory component, multisensory integration actually is not superadditive in C57 mice and emerges with age. So it's something that develops with time normally and fails to do so in these autistic mice. This is a feature of the auditory component. So if you look at across age, the size of the auditory field, both peak in the Black 6 mice and the BTBR mice, but clearly fails to prune in the BTBR animals compared to the C57. The tactile response is similar throughout ages, interestingly. So because of this, we decided to try and rescue these animals with diazepam during this early critical time when pruning should be happening and compare it to benzodiazepine or diazepam treatment in adulthood. And what we found was that there is a critical period when inhibition needs to be enhanced. We can inject animals with diazepam during this early pruning, normally early pruning period, or in adulthood for similar duration. And what we find is that we can make a BTBR mouse, which treated with vehicle would retain this large auditory field, prune away its auditory component in the insular cortex. As a result, the multisensory integration is rescued if you do this early benzodiazepine treatment. but not rescued by adult benzodiazepine treatment of similar duration. So there's something important about tapping into that early window. And in fact, if you look at the inhibitory markers in the insular cortex, you find that this early diazepam treatment actually promotes the maturation of GABAergic circuitry, as measured here anatomically, for example, with GAD65 staining or perineuronal nets. And so, given this result with this peculiar mouse, the BTBR animal, we also tested a number of other monogenic mouse models of autism and found interestingly similar results. That as this excitatory-inhibitory balance matures, in particular the parvalbumin cell function, this multisensory integration emerges. And so here you see the normal profile of development in young to adult C57 Black 6 mice. And in this case, if you enhance inhibition further acutely with benzodiazepines, you now inhibit the multisensory integration. There are a number of animal models that have autistic features because of this reduced inhibition, and they, in fact, fail to show this multisensory integration. I told you you can take a BTBR animal and fix it by giving early boost of inhibition, which then is translated into an enduring improvement. And there are also models on the opposite end of this spectrum. A few years ago with Michele Fagiolini's group, we were able to show that MECP2 knockout mice, which is a mouse model of Rett syndrome, show a hyperconnectivity of parvalbumin circuits, and these animals also show an impairment of multisensory integration in the insular cortex. And so there seems to be this inverted U-shape response of processing multisensory information, which in particular may reflect parvalbumin cell function. This is interesting to us because parvalbumin cells shown here with Brainbow technique are very pivotally poised. As we image in and out of this stack, you'll see the beautiful baskets forming around cell bodies. And some of these cells are parvalbumin cells themselves, and others are pyramidal cells. These inputs are poised to control the integrated output of their target neurons. And in fact, we can use them as a biomarker for plastic windows in the brain. Parvalbumin cells mature at different rates in different brain regions. We heard some very nice talks yesterday about how they migrate and arrive into the appropriate layers. And in fact, they develop in register with critical periods of plasticity. So for instance, we've worked on the visual system, which I'll turn to now, for quite some time. And we know that manipulating parvalbumin cell maturational state will in fact manipulate when this window happens. If you look in higher order mammals, as David Lewis has done in monkeys, you'll find that parvalbumin cells mature at later and later stages of the pathway, at later and later stages of development. And even within the mouse, earlier development of parvalbumin cells is seen in, say, the auditory cortex, predicting an earlier critical period there. So to test that, you can take a mouse and raise it in an acoustic environment where they hear a particular tone repeatedly, and then map the tonotopic frequency distribution in primary auditory cortex. And normally what you would see in single unit recordings from the primary auditory cortex, again delineated by these blood vessels, is that there's a nice rainbow spectrum of sound representation from low to high frequency across the surface of the cortex. If you raise an animal for just 3 days during an early critical period, around postnatal day 12 to 15, listening repeatedly to 7 kilohertz, the lime green color here, you get an expansion of that map at the expense of neighboring frequencies. And these maps are taken actually in adult animals long after the exposure, indicating the enduring nature of this early life experience when plasticity is particularly high. These changes are genuinely occurring at the thalamocortical stage, even though the auditory system goes through multiple relay stations, each one with a tonotopic map from the ear to primary auditory cortex. If we record from one station earlier in the auditory thalamus, no such changes are seen by this 3-day tone rearing. So in fact, this seems to be a plasticity happening at this last stage. And the thalamocortical plasticity, of course, is quite familiar to us in the mouse model or in animal models of amblyopia, as you've heard about in the first talk. Changes occur at this synapse. So In the case of mice, you can record their visual response directly from V1 and find there's a beautiful visually evoked potential which disappears as the sensory stimulus gets ever finer, giving you a static acuity of 0.5 cycles per degree in mice. Recalling that parvalbumin cells mature later than in the auditory cortex, there is a critical period around postnatal day 20 to 40 when patching an eye causes an imbalance of sensory input, and as a result, the acuity through that eye is enduringly decreased. And this is an experiment where the patching was done during the critical period, the patch was removed, and the acuity measurements are made in adulthood. And you find that there's an enduring effect on vision. Similar patching in adult animals doesn't produce this change. So, as predicted, the critical period for visual acuity is now staggered after that of the auditory system. This is, of course, an animal model of lazy eye, or amblyopia in humans, which has an even longer critical period, and as Tommaso mentioned, this may be related to the longer lifespan of ourselves, where the first 8 or 9 years of life is important, and during that time plasticity gradually winds down, leading to changes in the cortex as a function of imbalanced sensory input. The mouse allows us to test molecular mechanism, and that's what I'd like to focus on for the rest of the talk. Over the years, we found that manipulating the inhibitory half of this EI balance can actually turn the plasticity window on earlier or later. And on the flip side, once this is turned on, the closure of critical periods, as you heard in the first talk, seems to reflect the onset or expression of brake-like factors. And so I want to dig into this a little bit now. So one very potent consequence of monocular deprivation is an anatomical rewiring of input to the visual cortex. Work pioneered by Hubel and Wiesel and their progeny has shown that left eye and right eye input typically innervate the cortex in a one-to-one fashion, and that monocular vision for a few weeks of time actually causes a structural change, making the open eye inputs largely take over the visual cortex. This is correlated with a sequence of changes that was mentioned in the first talk as well, related to spine motility increases, leading to eventually spine pruning, and then spine regrowth and sprouting of axons. We were curious, though, that this takes so long. If you patch an eye, it takes at least 4 days to reach a point where you have a significant change in the spine number or density. Spine motility increases as well, but that too takes a couple of days of monocular deprivation. And so it seems like there is an earlier detector that is being missed by these studies. And we wondered, could this be the inhibitory neurons? Inhibitory neuron plasticity in this paradigm had not been studied despite the decades of work since Hubel and Wiesel. And so by knowing that parvalbumin cells might be particularly interesting, we could target recordings to these cells by their electrophysiological signature. Unlike pyramidal neurons, parvalbumin positive basket cells are fast spiking neurons. And so we can go in with a sharp electrode and compare the response to deprivation in regular spiking pyramidal neurons, which show a contralateral bias, more spikes to the opposite eye than the ipsilateral eye normally. And with monocular deprivation of the opposite eye, you see a shift or a loss of that contralateral bias at 3 days to 2 weeks of deprivation. It wasn't known whether these fast spiking cells are plastic at all and how they respond to left eye or right eye input. And in fact, it turns out these cells are equally innervated by both eyes normally, and left eye, right eye input drives a similar amount of spiking. With monocular deprivation, they are very plastic. In fact, they shift their response in favor of the closed eye first, and then in favor of the open eye later. This result has been replicated in the cat model, the traditional model. This is data from Marcos Frank, who was recording from the visual cortex of freely behaving kittens. And in the cat, a much more visual creature than the mouse, plasticity is faster. And so already within 6 hours of monocular deprivation, you see the same thing, fast spiking interneurons shift in favor of the closed eye, and then later in favor of the open eye. and this actually preceded the change seen in the pyramidal cells. And so this started to suggest that maybe the importance of these neurons for critical period plasticity is that they are the first responders to monocular deprivation. And we started to test this idea at higher resolution. In order to do that, we had to develop a thalamocortical brain slice preparation. Cutting the brain at a funny angle preserves the connection from the visual thalamus here, wrapping around its connections up to visual cortex. I hope this little movie will work. If we soak the slice in a voltage-sensitive dye, you should see a puff of activity when the stimulus is applied to the LGN, and then a response some milliseconds later in the visual cortex there. Okay, and so using this slice, we can then stimulate the visual thalamus and patch onto neurons in the binocular region of visual cortex and see what is the consequence of monocular deprivation. The first thing you notice is that parvalbumin cells are very potently driven by thalamic input. We stimulate at low intensity in the visual thalamus. I'll show that here. By placing a stimulating pipette at different sites along the LGN. And in fact, you can pick up a lower stimulus threshold when you hit cell bodies in the ipsilateral patch, as opposed to stimulating fibers exiting the bundle here from the LGN. Using this criterion and then the first all or none response in the cells we're recording from, we can find that parvalbumin cells receive an overwhelmingly stronger response directly from the thalamus with this significant conduction delay. Non-PV, meaning pyramidal cells, show a small response. This connection is already present before the critical period in both cell types and is always more powerful in the parvalbumin cell. If you look at a number of parameters like the onset delay, the reliability of the response, how much jitter is there when stimulating the thalamus, the size of the response, and related to that, the pair pulse ratio and AMPA receptor kinetics, you see that the PV cells are poised to receive thalamic input and be very sensitive to changes in this connection. And in fact, short-term monocular deprivation for a couple of days produces no change in the thalamocortical input to pyramidal cells, but it has a dramatic effect on parvalbumin cells. This is a particularly strong example. On average, you see at least a 50% reduction in the size of the EPSC driven from the thalamus in the parvalbumin cells, whereas the pyramidal cells don't change at all. And so in studies that have followed up, we find that this change can be elicited even 2 hours after monocular deprivation has started, certainly well before the days of time it takes to change input onto pyramidal cells. This seems to be a presynaptic change because paired pulse ratios, as you can see, clearly change onto the PV cell. Is it relevant to critical period plasticity? Yes, we think so, because if we do monocular deprivation in the pre-critical period or in GAD65 knockout mice, which never enter this critical period, this thalamocortical change is not seen on the parvalbumin cell. We looked anatomically then, using this VGLUT2 marker of thalamocortical terminals that Tommaso mentioned, and looked at the puncta they form onto the cell bodies of parvalbumin cells, and found that, in fact, the size of these presynaptic terminals changes significantly onto parvalbumin cells, and this precedes an actual reduction in the number of puncta. And interestingly, we also noticed a shrinkage of the target cell body size. And so this starts to give us some idea maybe what's going on here. There is clearly a presynaptic impact of monocular deprivation, which occurs fast and first onto these cells, and it has consequences on the cell state of the parvalbumin cell. This reminded us, of course, of work that we had started with Alain many years ago, indicating that the visual pathway sends not only action potentials, but perhaps molecular messengers which maintain the maturational state of parvalbumin cells. This, of course, is OTX2 that I'm talking about. And OTX2 may have a kind of morphogenic, loosely stated, function in parvalbumin cells. And with monocular deprivation, due to the reduction of presynaptic input, these cells may suffer. The perineuronal net, which surrounds these parvalbumin positive cells, may act as a kind of trap or a sink for OTX2. And this is because the OTX2 sequence contains motifs which are identified in sugar-binding proteins. And in particular, this short peptide sequence, which includes an arginine-lysine doublet, is found. And in work done here in France, the RK peptide sequence is sufficient to bind to sugar motifs in the perineuronal net. If you scramble this peptide to retain the charge but divorce the R from the K, you lose this affinity. And if you mutate RK to a double alanine, you certainly lose that affinity. For us, this represented an excellent tool. This short peptide sequence could potentially be injected in vivo to compete with sugar binding sites with endogenous OTX2. And in fact, if you infuse RK peptide in vivo, you can knock down the accumulation of endogenous OTX2 in the visual cortex. That's not seen with scrambled peptide or this AA. So what happens to parvalbumin cells specifically? If we inject RK peptide and a couple days later look at the parvalbumin cells in PVGFP mice, you find that they've lost their OTX2 content, replicating what I just showed you. And it clearly impacts their physiology. So these cells, as I said, are fast spiking neurons. This is a property which actually matures further as they get older. And so in these FI plots where we inject current of increasing strength, you find that in color from orange to yellow to green, that as the animals mature from eye opening to pre-critical period to adulthood, this fast spiking property gets better. And if you inject a vehicle into adult animals, nothing changes there. But if you inject RK peptides, you find that the fast spiking property is reset to a just pre-critical period state. And what about synaptic inputs? Here's an illustration of what we think is going on. The perineuronal net is perforated by synaptic boutons. And if we're actually resetting the PV cells to a juvenile state, what happens to those inputs? Well, in fact, we can decrease the number of excitatory inputs onto PV cells. Looking at spontaneous events here, you find that the amplitudes of the less frequent inputs are only the ones that are the strongest. And so there's a net shift in favor of stronger amplitude but far fewer input onto these rejuvenated cells. And under these conditions, you can reopen the critical period. So in adult animals treated with this RK peptide, visual acuity, which normally would not change with monocular deprivation at this age, can change. And conversely, if you make the animals amblyopic deliberately, treating them with RK peptide actually restores visual acuity. Interestingly, in this particular experiment, we didn't use RK peptide. We knocked down OTX2 at its source, which I think Alain will elaborate on, includes not just the visual pathway, but the choroid plexus, which is a kind of global broadcast center for this OTX2, and in fact might explain why this factor is relevant not only for visual cortex, but for parvalbumin cells throughout the brain. So the model we proposed is that just like the EI balance being delicately poised in an inverted U shape for proper brain function, plasticity control is also controlled by a two-threshold model, and that it reflects the maturational state of parvalbumin cells. In the case of the cortex, at least, it's sensitive to a gradient of OTX2 building up inside these cells. You need a certain amount of OTX2 for these cells to engage the plastic process, and they keep accumulating this molecule, and we're very much interested in the targets once inside the parvalbumin cell. Amongst them, it seems there's a positive feedback to produce more tight nets, and as a result they continue to accumulate OTX2 and pass eventually this higher threshold, which keeps the plasticity off. And so it seems like a rather complicated system to regulate when and where plasticity should happen. You might wonder, why do this? Plasticity seems like a good thing. Why turn it off? And I'll finish by saying that, in fact, we can turn it off by these manipulations. And this suggests that there's a reason for keeping it off at some cost, producing these nets throughout our adult life. And we think the manifestations of not turning it off can again be seen in mental illness. Perineuronal nets are late developing structures, in this case work from Sabina Beretta, looking at the prefrontal cortex of humans. You find that these nets emerge slowly in this last to develop brain region over several years. And this is in so-called neurotypical controls. I'm not quite sure what neurotypical means, but interestingly, if you compare that to patients who had schizophrenia, this is a case where perineuronal nets are compromised and provides perhaps an opportunity to understand why it's important to close out these critical periods. Last year, we published a paper with Kim Do's group where we focused on another function of these nets, and that is as a buffer against reactive oxygen species and oxidative stress. In an animal model which is undergoing early stress, we find that parvalbumin cells that have no perineuronal net, little or no perineuronal net, show greater signs of oxidative damage than parvalbumin cells who do have a net. And it suggested that there may be this inverse relationship, that it's a neuroprotective covering as well. And if, in fact, if we deliberately remove the nets, then we can make adult parvalbumin cells quite vulnerable to oxidative stress. Since oxidative stress is a kind of hub in schizophrenia mouse models, at least, we decided to test this idea by deliberately generating animals in which parvalbumin cells specifically are devoid of their primary antioxidant system, glutathione synthesis. You can do this by knocking out conditionally the GCLC, the catalytic subunit of a GCL, only from parvalbumin cells. Thanks to the parvalbumin-Cree line of Sylvia Arbour, we see that this is a progressive loss with age. And in fact, this causes an increase in oxidative damage in parvalbumin cells selectively. As a result, these cells suffer, and parvalbumin cell boutons are decreased, and eventually perineuronal nets also are compromised. They're a protective shield, but under extreme oxidative stress, they too are compromised. And so what would you predict from this? The consequence of early life stress, something very bad to the brain, actually improves brain plasticity. So the loss of this perineuronal net allows these adult animals to show monocular deprivation effects. And it's specific to the parvalbumin cell, because if we produce GCLC deletion in pyramidal neurons, not the inhibitory neurons, we don't see that effect. And so something about the maturational state of parvalbumin cells is extremely important for controlling when plasticity turns on and off. So a fanciful idea is that perhaps, at least during some stage of neurodevelopment, a risk factor for schizophrenia might include too much plasticity, which would suggest alternative kinds of therapy. This actually makes sense if you look at review articles of prefrontal cortex and how this prodromal phase is played out. You see signs of excessive plasticity as revealed by deficient myelination, perisomatic inhibition, and excessive spine pruning. So this is what I told you today. The excitatory-inhibitory balance is dynamically changing during development. A number of factors are important for this. And once this balance matures, circuits become plastic. On the flip side, this plasticity is controlled by the additional layering of brake-like factors. Today I only had time to talk about one in detail, which is the perineuronal net. Myelination is another clear contributor, epigenetic changes, and all of these can be reversed in experimental settings anyway by injecting something into the brain. There are other breaks that act on neuromodulators, our neurochemistry in the brain which allows plasticity in adulthood, and all of these things may be allowing us to gently restore this excitatory and inhibitory balance which lives in that narrow range for plasticity to occur. And so we know that this is an interesting possibility. Are there any French speakers in the room? So if we can actually harness this capability, then as Tommaso was illustrating, maybe we can repurpose this for therapeutic approaches. This is an idea which has incredible resonance with the public. We published a proof of concept paper where we did something silly to try to see if we could introduce critical period type learning like absolute pitch, which needs to be acquired typically before the age of 7 by opening such breaks. This paper was published in December and within 1 month had almost 100,000 views. You can imagine the hysteria that could ensue. We're not interested in doing that. We want to use it as a therapeutic tool. So an approach that we're taking is to modulate neuromodulatory systems to recover from amblyopia. This is something that's also being done in Europe by Eero Castran and Lamberto Maffei. So we talked about excitatory inhibitory balance, and it's influenced by many factors. And this is why the study of critical periods and brain plasticity is so challenging. Documenting all of these environmental and genetic factors is going to be a big challenge for any individual human. Okay, I will stop. The work was done by many people. The Insular Cortex project was work by Nadine Gogola, who's now back in her own lab at Max Planck in Munich. Parvalbumin cells and inhibition has been the workhorse of our laboratory and has been greatly spurred by collaboration with Alain. The auditory critical period work was done by Tanya Rinaldi, who has her own lab in Denmark, and human studies related to that with Janet Worker. And the oxidative stress and other molecular breaks on critical period closure was work done by Hiro Morishita, who has his lab at Mount Sinai, collaboration with Kim Do, and the clinical trials with David Hunter at Children's Hospital. And this is ongoing with people at Harvard. Thank you very much.